Now that we've looked Now that we've looked at glycolysis and gluconeogenesis, I want to look at the citric acid cycle because the pyruvate, the product of glycolysis, can then be further oxidized so that its carbons can enter the citric acid cycle. To be clear, citric acid cycle is exactly the same thing as, to be clear, other terms which mean exactly the same thing as a citric acid cycle is Krebs cycle, tricarboxylic acid cycle, and TCA. Tricarboxylic acid because a number of the intermediates in the citric acid cycle have three carboxylic acid groups on them. Just like with glycolysis and gluconeogenesis, I don't want you to memorize a single enzyme name, but I want you to be able to interpret every one of those enzyme names. In particular, you should be able to identify the substrate in the product based on the name, maybe have some idea of how it fits into Krebs cycle. Also, there's a lot of movement. Also, a number of the steps in Krebs cycle involve oxidation driving a reduction, so you should be able to know what substrate or product is oxidized, which one is reduced, which undergo no. Which substrate undergoes an oxidation or a reduction, what goes up in energy, what goes down in energy. And there's a very nice chapter in the Lippincott textbook that describes that very well. Again, comparing this to the glycolysis and gluconeogenesis talks, we mostly focused on the intersection between glycolysis and the pentose phosphate pathway and glycogen synthesis. We mostly looked at the enzymes which connect glycolysis to the pentose phosphate pathway, or to gluconeogenesis, or to the pentose phosphate pathway. So in looking at the citric acid cycle, I would like to look at what connects glycolysis to the citric acid cycle, and the focus is going to be on pyruvate and on acetyl coenzyme A. And so pyruvate can have 4 different fates in the cell. Pyruvate has 4 fates in the cell. Can you list the compounds which pyruvate can be converted to? And can you list the compounds which can be, will you list the compounds which can be formed by a single chemical reaction, which can be formed out of pyruvate through a single chemical reaction? And for each of them, list the number of carbon atoms. It turns out the number of carbon atoms is going to help you sort things out. In fact, the information I'm asking for is the information I'm looking for is right here in the figure, so go ahead and fill it out, but I'm going to move on. So pyruvate can be converted to alanine with a simple reaction. Pyruvate can be used to make acetyl coenzyme A. Pyruvate can be converted to lactate, and pyruvate can be converted into oxaloacetate. Pyruvate can be converted to alanine. Now, can you tell me what type of reaction? Well, in going from pyruvate to alanine, you should know, I think you know this from the amino acid pre-matriculation talk, that pyruvate can be converted to alanine by exchanging the ketone for an amine to make an amino acid instead of a keto acid. Or alanine can likewise be converted into pyruvate by a deamination or a transaminase reaction exchanging between the amine and a ketone. Or pyruvate can be converted to lactate. Do you know what enzyme does that? We've looked at it a lot. How about lactate dehydrogenase? And again, that tells you about the energetics and about what cofactor and about what coenzyme you think should be used. And then, of course, pyruvate can be converted to oxaloacetate, or it can be converted, or pyruvate can be used to make acetyl coenzyme A. Why do you think I colored two of these arrows red and two of them green? Please pause and answer. Two of the reactions are reversible, as indicated in green. Two of these reactions are reversible, as indicated in green, with arrows on both sides of the line. And two of these reactions are irreversible. There's a drop in energy so that they cannot go back uphill, and that's indicated with the red arrows. Now, can you name each of the four enzymes responsible for these reactions? Please go ahead and do so. And for that matter, can you tell me what the product will be used for? And for that matter, will you also tell me what the product will be used for? And for that matter, will you also list possible uses for each of these products? Please go ahead and do so. It's in your interest to do this right now, and I think you can simply by interpreting enzyme names. Well, of course, if you convert pyruvate to alanine, well, you have an amino acid. You could use it to make protein, or you can be converting back and forth between lactate dehydrogenase. Lactate dehydrogenase allows the interconversion between lactate and pyruvate, and this is really useful in sharing metabolism between cells. For example, your red blood cells build up a bunch. For example, your red blood cells are exclusively anaerobic and make a lot of lactate, which they release into the blood. Then the lactate can be filtered out by the liver, converted back into pyruvate, and sent into gluconeogenesis so that the red blood cell can share its anaerobic metabolism with the aerobic metabolism of the liver. Pyruvate carboxylase. Now this is a really important distinction. It's real important to know the difference between lactate dehydrogenase, pyruvate dehydrogenase, and pyruvate carboxylase. The names tell you exactly what they do. An important distinction is that pyruvate carboxylase is the first step of gluconeogenesis. So that's important. It's also important to know that pyruvate carboxylase is also used to replenish citric acid cycle intermediates. So let's say that you're really low on sugar, your brain needs sugar, and you're running a whole lot of gluconeogenesis in the liver while you're converting oxaloacetate into sugar. And that can deplete the intermediates in the citric acid cycle. In order to replace them, you would want to use pyruvate carboxylase, which takes pyruvate, which is not a citric acid cycle intermediate, converts it to the 4-carbon oxaloacetate, which is now a new citric acid cycle intermediate. Let's compare pyruvate carboxylase to pyruvate dehydrogenase. Pyruvate dehydrogenase is used to make acetyl coenzyme A and carbon dioxide. Then acetyl coenzyme A can donate its 2-carbon acetyl group to oxaloacetate to to make citrate. So it has increased the number of carbon atoms on oxaloacetate from 4 in oxaloacetate to 6 in citrate, and now you can burn it. But because you are simply converting one citric acid cycle intermediate into a different citric acid cycle intermediate, you've not changed the numbers. You've not changed the net number of intermediates. So that is, pyruvate dehydrogenase cannot be used to replenish the number of citric acid cycle intermediates. If you're low on citric acid cycle intermediates, you need to use pyruvate carboxylase, or you might be able to convert some amino acids into that also. And that's an important enough concept that I describe it here. In addition, this pyruvate carboxylase, which takes a 3-carbon pyruvate and makes a 4-carbon oxalic acid, is important enough that I want to show this in a figure, that I don't just want to point out the reaction and give you the name of the enzyme, but I want to actually show the reaction because I think seeing it visually will help you remember it.